and welcome to your next section on evidence for evolution. So we left off talking about fossils and how they were involved in evolution, but now I want to take you through more parts of evolution. So our third set of evolution evidence, this is going to start out with biogeography. Biogeography is the geographic distribution of species suggested to Darwin that organisms evolved from one common ancestor. Now, when Darwin went to the Galapagos Islands, he saw these adaptations. That's where he got the idea for natural selection. So he went back through his notes and he was able to note that animals on islands resemble species of nearby mainland more closely than they resemble animals on similar islands and other continents. This is because they are evolving from one common ancestor and they are not evolving from each other. So, our next evidence for evolution is comparative anatomy. Now, I know we've talked a lot about this in class. You're going to get a better understanding of it here. So, comparative anatomy, in its truest sense, is the comparison of body structures in different species. Now, we looked at homologous structures. We talked about homologous and analogous structures. We're going to go through a little bit about what each of those are. So homology is just the similarity in characteristics that results from common ancestry. So having a common ancestor is going to give you those similar characteristics. Now, when we look at this, we talk about the homologies in vertebrate forelimbs and how bone structure is so closely related in all of these vertebrates. So some homologous structures, though, are vestigial structures. Now, I know we talked about our appendix being a vestigial structure. If you didn't pick up on it previously, vestigial structure are parts of our anatomy that were once useful to our ancestors but are no longer useful. So moving on, these are actual homologous structures. You see, we have a human, we have a cat, we have a whale, and we have a bat. And you can see the different colors, and you can see all of these have a humerus. And that humerus is different shapes for the different structures. So it's evolved to be used for those structures. All of them, you can see the radius and ulna. Some of them are fused, some of them aren't fused. And then they have the carpals, metacarpals, and phalanges. Now in homologous structures, you can have the same use, kind of like a chimpanzee and ourselves, or you can have separate uses like the whale and the cat. So we talked about homologous structures. What about analogous structures? We talk about analogous structures when we talk about evolution, just to touch on that they do not come from a common ancestor. They have the same function, but they do not come from a common ancestor. The analogous structure would be like a bat wing and an insect wing. They're both used to fly, but yet the ancestry is different based on the structure. So our next evidence for evolution is the fifth evidence, and that is comparative embryology. Embryology meaning the study of development of organisms. So comparative embryology is the comparison of early stages of development among different organisms. So we're just looking at that embryo and seeing the difference within it. And many vertebrates have common embryonic structures revealing our homologies among them. A lot of times vertebrates look very, very similar as embryos, and then as they grow, their genetics give them the different structures and characteristics that makes them whatever animal they may be. So when you were an embryo, you had a tail and you had pharyngeal pouches. This is exactly like a fish, a chicken, all kinds of different vertebrates. So we are very similar in our embryonic stage. Let's move on to look at some pictures of this. So this is a comparative embryology example, and as you can see, you have a fish, you have a turtle, a salamander, and a human. And as you can see, all of these start out with a tail, even you do. and they have the pharyngeal slits, as you can see right through here. And as you grow, you lose that tail and you lose those slits and you become who you are today. But the fish uses that tail, it develops into its fins, and those slits develop into its gills. So we can see, based on this embryology, that we are very similar in our ancestry. Okay, our next evidence for evolution is molecular biology. Now, I know we've talked a lot about molecular biology. We just finished a unit on molecular biology. So some of this might sound a little repetitive based on that, but it'll make sense based on you knowing that molecular biology. So molecular biology is comparisons of DNA and amino acid sequences between different organisms, which reveals our evolutionary relationships. So we're using that DNA, those amino acids that we do in transcription and translation, and we make them, and we're looking at them to see what the actual differences among our amino acids and DNA are. So all living things share a common DNA code for proteins found in living cells. So you share genetics with bacteria, yeast, and even fruit flies. But when we look at this, we look at an evolutionary tree. And we're going to take into account all 6 of those different evidences for evolution. And as you can see, this evolutionary tree is going to show you how we are related to each other. We start out here at this number 1, where we have a common ancestor. All of these numbers show the common ancestors. And where you have these tick marks, it's a homologous characteristic. So at this first tick mark, tetrapod limbs came into play. Then at this tick mark, amnion came into play. So those eggs that weren't dried out due to the amniotic sac came in. And you can see feathers as well. So we go down from a lungfish all the way to a hawk or other birds. So we are all related some way, somehow, by this common ancestor right here. Okay, so we've talked about all the evidences of evolution. I want to remind you of a few things. We're going to go into a little more detail, and then you will have another video on more types of evolution and how we use these later on this week. So populations are the units of evolution. Now remember, a population is a group of a species that live in one area. And remember, we talked about how populations are the unit that evolves. Individual species do not evolve. So a gene pool is the total collection of genes in a population. And remember, when we have variation, we can have natural selection. If we do not have variation, we do not have natural selection. Remember those 4 pillars of evolution. We're going to kind of refer to those as we go through these. So microevolution is changing the relative frequencies of alleles in a gene pool over a period of time. So we're looking at those alleles, we're looking at those frequencies, that genetic variation, and it's going to change over time. And that's going to give us the differences that we have. It's not necessarily that adaptation, it's actually changing the genes that are available. So population genetics studies how each population changes over time. And remember, our populations are what changes, not our individuals. It's a pretty important fact. Okay, so if we talk about mutation and sexual reproduction, this is what gives us our genetic variability. Without sexual reproduction, we are the same. So remember from meiosis that sexual reproduction we have the crossing over, we have all that genetic variation coming into play when we make our gametes. And the formation of the gamete is what makes the individual organism. So mutation, or the changes in the nucleotide sequence of DNA, is the ultimate source of our new alleles. So mutation is what is giving us this genetic variability. And occasionally, mutant alleles improve the adaptation of an individual to its environment and increase its survival and reproductive success. So not necessarily is a mutation a bad thing. A lot of times we talk about a mutation as a bad thing, but this can actually increase the fitness level of the organism for its success in its environment. So when we talk about mutation, it can be good or bad, and some mutations are very good for these organisms. Okay, so moving on in mutation and sexual reproduction, I just told you a little bit about how we combine and went over meiosis a little bit. This is going to go over meiosis a little bit more, so it's going to be a little bit of a review from when we did our genetics unit, but it's good for you to review. So sexual reproduction shuffles these alleles to produce new combinations. Remember, all of that meiosis. So we have those homologous chromosomes. They sort independently as they separate during anaphase 1 of meiosis. So remember that law of independent assortment? This is really coming into play with evolution. So during prophase 1 of meiosis, pairs of homologous chromosomes cross over and they exchange those genes. Remember that crossing over, we're going to have one piece of a chromosome attached to another piece of another, and that's going to give us more genetic variability. So this is where we really see the change in our populations from this sexual reproduction and meiosis. And further variation arises when sperm randomly unite with eggs in fertilization. There is no way we can choose when we naturally fertilize an egg what sperm and what alleles match that egg. And we cannot choose the egg which is being chosen to be released either. So it is all random. So all this randomness gives play to our genetic variability, which makes adaptation easier for our environment, which lends to evolution. Okay, this is all you guys have for today. Remember, you have some homework other than this weekend, and we will be looking at these notes on Monday. You will have another set of notes on Tuesday, so just stay tuned for your next video lecture.